Discovering the effect of Senna alata on the expression of cep-1 in C. elegans, presented by Isabelle Hoover, Zainab, Soofi, and Josh Rizer. Cancer is not only a prevalent disease, but its treatments tend to be inaccessible and expensive. Because of this, we wanted to look for different supplements that can help individuals fighting cancer. Our hypothesis is if C. elegans are exposed to a diet incorporating the Senna alata plant, then they will have a higher expression of the cep-1 gene. Our question asks, if we expose varying amounts of Senna alata on cep-1 reduced C. elegans, will the expression of the cep-1 gene increase, decrease, or remain the same? And behaviorally, will the result in the thrash count population rate increase or decrease, indicating a health change? Senna alata and similar homeopathic plants have anti-cancerous effects due to their phytochemicals and are already used medicinally, making them very accessible. C. elegans are used as the model organism because of their tumor-suppressant homologue cep-1 and identifiable thrash count. We used a mutant that has a depleted cep-1 gene called TG12 from Minnesota University to test the gene's increased expression. We begin with making our concentrations of Senna and prepare our E. coli and C. elegans in the plates. We then test their thrash count and other qualities and move into PCR. Afterwards, we will soak them in bleach and dispose. qPCR specifically begins with RNA extraction and then gathering and storing our cDNA before actually beginning the qPCR to determine cep-1 expression. We first tested Senna alata's effect on E. coli, and we saw some antibacterial properties, but our p-value led us to believe there wasn't much significance. From there, we could move to applying our C. elegans and collecting their thrash count data. We found that thrash count was relatively the same in our wild type, but significantly increased with our mutants the more Senna we applied. We ran an ANOVA test and found a p-value less than 0.01, which means the data is statistically significant and we can believe that Senna increased the health of our mutant type C. elegans. By incorporating Senna alata into C. elegans' diet, we were able to increase their cep-1 expression and health. We hope to corroborate this with qPCR data and apply it pharmaceutically.